All right, hello. Hello, who is?
Introduce you.
Who? Yeah, you are?
I'll do that.
Okay, I'll go down. Oh, no, it's okay.
You want this thing?
I'm fine, just go ahead.
So good afternoon, good evening everyone. It is wonderful to see an overfull house today. People on the, right. Well, the assumption is that the talk is going to be so great. Everybody is here for the talk. And that is always wonderful. And it is indeed a pleasure and privilege to introduce Professor Utpal Banerjee, who is a distinguished professor in the Department of Molecular Cell and Developmental Biology at the University of California, Los Angeles. He did his master's from IIT Kanpur and then PhD at Caltech, and after a few postdocs joined UCLA and has been there for quite a long time. He is certainly, from what I can understand, a prominent figure in the area of modern genetics and has done spectacular groundbreaking research into the fundamental signaling pathways that govern development in living organisms. And he is also equally well known for very path-breaking advances in undergraduate education at UCLA also. So his scientific contributions are in the area of sort of decoding the language of cells using mainly the fruit fly as a model organism, from what I understand. And he had done some of the pioneering work on various signaling pathways that control cell growth and differentiation. From what I have read, Professor Banerjee's work has also revealed ways in which sort of genetic instructions can arise or get modified by signals from outside, which is something important for understanding things like cancer, which afflicts human beings. It's also made seminal contributions in metabolic regulations and development of blood cells and so on. List is quite long. And not only he is a member of American Academy of Arts and Sciences and National Academy of Sciences of US. Professor Utpal Banerjee is also an award-winning teacher. Yes, pioneered the undergraduate research consortium in functional genomics, which is a program in UCLA under which UG students learn by essentially doing research. So it's a discovery-based sort of pedagogy, which is something that all our students here at IISERTVM can resonate to, because that's also our mission, to put undergraduate education in a vibrant research setting. And he has a long list of recognitions, some of which include the NIH Director's Pioneer Award, the Elizabeth W. Jones Award for Excellence in Education, Howard Hughes Medical Institute Professor. He has been thrice, which carries a large research support of $1 million or so with it. He's also been honored with the Distinguished Alumnus Award of IIT Kanpur and the top 20 professors of the Bruin Century, which is in UCLA. And so it's definitely a pleasure and honor to welcome Professor Banerjee to deliver his colloquium today at IISERTVM. Well, the title is Metabolism Drives Pre-Implantation Embryonic Development, but the one that we had circulated in the poster also said Developmental and Cancer Metabolism, an introduction for future physicists, chemists, basically everybody in IISER. So with that, I would like to invite Professor Banerjee for his talk.
Thank you. All right. Thank you very much. Thanks for the invitation. Thanks for everyone for your kind hospitality, which I have enjoyed a lot. If you would turn these lights off, it will help some of the later slides. Okay. As you just heard from the introduction, my lab over 30 years has been working on finding various signal transduction pathways in Drosophila. And you know, going back many years, it has got to do with finding the EGF receptor equivalent and finding son of 7-less, which is the first GEF, the exchange factor that was found, and then you know, signaling pathways together. But today I'm going to talk about something quite different. Obviously, you see this pre-implantation embryo, so it has to be a mammalian system. So this was a sort of a short stint to work outside of the fly, and the work, all the work that I'll describe, was done by these three guys. Raghavendra Nagaraj, I sort of recruited from TIFR some 25 years ago or something like that. He pretty much retires with me. Mark Sharpley is a professor at Cedar Sinai Hospital, and he's the metabolism person. And Feng Tao Chi is probably my best graduate student ever. Let's get rid of this. Who has worked on a lot of different parts of it. So I'm not going to keep telling you who did what, because there were only three, and it'll become, and they did all of it together. Now, the title requires, I did change the title, it requires a little bit of an explanation, because it says metabolism drives pre-implantation. It doesn't say it allows it. It's understood that every cell requires a metabolic pathway to do something or the other. Can you hear me? Yeah. So, but then driving a pathway is something that is come rather late in our studies, in anyone's studies. Because if you think about it, when it came to cancer, the chemists had, biochemists had components of various MAP kinases and transcription factors and so on and so forth. And the cancer people had the cancer version of it. But what really bridged the gap, where you know many of us came in, was trying to understand what the gene function was. Because a person doesn't go into a doctor's office and say, you know, my so-and-so gene is not functional. So that was the development part of it, which was done largely in model systems. Now, when it came to metabolism or metabolic pathways, it sort of skipped that step. The chemists, biochemists, again, took the lead role in finding all the components, all the enzymes, and so on and so forth, as they have done always. And then these genes were found mutated in certain cancers. What is a metabolism gene, mitochondrial gene doing in a cancer situation is what drove a huge field into cancer metabolism. But what it did not go through was really the developmental metabolism step, which I want to concentrate on today. So what is developmental metabolism? It says that this idea that metabolism is only involves housekeeping genes, as they call it, which irritates me a lot, that it involves housekeeping genes which are allowing a system to work, and you know, whatever diabetes or exercise, etc., is of course important, but cellular development, going from one cell type to another cell type, there we have to go back to the signal transduction pathways that we have always talked about. Now, in our fly experiments, it was very clear that that is not 100% the case. I mean, I'll be the last one not to defend signal transduction, but we knew that metabolism can have very specific effects. And I hope to convince you of that today. Okay, so okay. This thing is like vibrating when I... Here, let me just control it there. We are running late already. Okay, it'll work, right? Guaranteed. Okay. All right. So, just a very quick sort of summary of all the metabolism that we really need to understand this talk. You know, I know this is an audience of all scientists, and so therefore I want to make sure that you see that there's nothing fancy that I'm talking about. Everything is very simple. Glucose is a primary source of energy, as you all probably know, for many, many different cell types. And the way glucose works is through these different steps of glycolysis, it then leads to pyruvate. Okay, pyruvate is the simplest ketone acid that you can conceive of, and that pyruvate has one of two options. The pyruvate can enter the mitochondrion, and then it is involved in running what's known as the TCA cycle, or the tricarboxylic acid cycle, also known as Krebs cycle, also known by various names. Now, or alternatively, that pyruvate can get converted into lactate by this enzyme lactate dehydrogenase. Now, in plants, under anaerobic conditions, this lactate can then be converted into alcohol. That's how the wine industry works, and thank God we don't have that gene, or we will be walking around drunk all day long. Okay, so this is all that we really need to understand here. Okay. Back. You go back? Okay. So, in order to understand this process, what does one do? There are various ways of doing this in Drosophila that I don't want to go into because it's a different talk. How do we do this in mammalian systems? A liver or a heart or a brown fat and so on, so those are too complex for me. Okay, I like to work on simple problems. And so the question is, where can I understand the role of metabolism, which is defined as anything that breaks a carbon bond, essentially, but metabolism in the process of development. And so after a lot of thought, I decided to concentrate on this particular process, where if you take some single cell embryos from a mouse or a human, any mammal, it goes through these repeated divisions, cell divisions, 2 cells, 4 cells, 8 cells, and then this is a compacted morula, it's called. It's a little more than 8 cells. And then this is the blastocyst, which is what has various cells as, from which the stem cells are derived, as well as from which the trophectoderm, which is the placenta and the stuff that will not make it to the animal, are derived. Okay, so we all know about the stem cells, which is this inner cell mass. Why choose this system? Well, first of all, there is a well-defined system of growing these cells. They will grow very nicely through this whole process, because the IVF people, the in vitro fertilization people, have already figured all this out. And they have added a lot of things to their media, which makes them not only go into this state, but then they can be transplanted to make an actual organism. So some of it is known, but our goal wasn't to find out what all you can add to the medium to make this process better and better and better, which is obviously what the IVF people would want to do. Our goal was to see what is the minimum you can get away with. And when you do that, you start taking things away from the medium, and you find that all you need is some salt and buffer. Okay? And these three metabolites, these three nutrients, which have been known for 100 years to be important for mammalian development, and they are glucose, pyruvate, and lactate. And pyruvate and lactate exchange with each other at a very fast rate because of lactate dehydrogenase being present, and then there is glucose. Amazing, right? This whole machinery works all the way up to the blastocyst. We took out all amino acids, all the lipids, everything that was in the medium, and this was still perfectly fine. Okay. Okay. Now, what is the function of a mitochondrion? Any student wants to speak up? And what's the function of a mitochondrion? Quickly, I don't have much time. Someone in the front, because I can't see in the back. What does mitochondrion do? Makes power, right, or makes energy, right? Now, I can tell in a mixed student group in the US, who is born in US versus who is from the outside of US by asking this simple question. No genetic tests needed. I just ask, what does the mitochondrion do? If you're born outside the United States, you say to make power or to make ATP and so on and so forth. If you're born in the United States, you say it's the powerhouse of the cell. Someone on television must have said that at some point of time, that it's a powerhouse. But it's a very simple test, requires no gels. Okay, now we know that is not just what the mitochondrion is required for. Because if that was all that it was required for, then we have glycolysis, we have other ways of generating ATP, we have anaerobic organisms probably outnumbering the numbers that use oxygen, and yet the power cannot be the only function because they all have mitochondria. So you know the answer to why, and that is because of the synthetic and the degradation pathways, what is known as anabolism and catabolism. So that's good. Now, amazingly, there is one bit that is that everyone knows, but it is not somehow emphasized, and this is worth thinking about. And that is that when we talk about epigenetic changes, we talk about proteins that are in the nucleus, which then are modified in some way or the other. But where does that modification come from? If you're working on epigenetic changes, you're working on the mitochondrion. You are working on metabolism, because OGT, okay, that's a complicated one, but SIR2 is utilized for NAD, histone acetyl transferases for putting the acetyl-CoA. I mean, the acetate group of acetyl-CoA does not come from somewhere. I mean, we don't just drink acetate in order to incorporate it, right? It is the mitochondrion which makes acetyl-CoA, which is then needed for all the histone acetylation that we can think of. And then there's more, TET enzymes. These are the demethylases, and that's also made from alpha-ketoglutarate, which is generated in the mitochondrion. Methionine is generated in the mitochondrion, and through SAM, it then is required for methylation purposes. And of course, this one we all know that ATP is generated in the mitochondrion, and that has got functions in epigenetics. So there's no such thing as studying epigenetics and not working on metabolism. Metabolism and epigenetics are tightly linked, because there will be no epigenetics if there was no metabolism. So, and therefore, even if you don't need it for energy, you will still preserve the mitochondrion. Okay. All right. So now this is again a repeat of the first slide that I showed you. It's 1 cell, 2 cell, 4 cells, and it's going from 20 hours of development to 114 hours of development with just those 3 metabolites present in the medium. Okay. Now, this is a result which we have obviously reproduced here, but the result has been known for almost 100 years. And who is who in developmental biology at some point of their lives have worked on these sort of very, very early embryos, and they have shown that a single cell, if you do not have pyruvate in the medium, the cell goes to 2 cell just fine, but then it stays at 2 cells until it dies. So it stays for a long period of time at the 2 cell stage, and this has been called the 2 cell block for many years. And this is minus glucose. So if you did not have glucose, the cells have no problems going through the 2 cell, 4 cell stages. They will work without glucose, but then when it comes to the 8 cell stage, they block and they do not go to the blastocyst. Very simple questions, why? For 100 years, people have been trying to figure this out. Now, why do we need pyruvate for 2 cells, and why do we need glucose for 8 cells? Now, the reason to choose this system in the first place was that mammalian embryos, being the only ones that are internally fertilized, are at this point of time, this is pre-implantation, at this point of time, they're floating around in a very tiny amount of fluid called the oviductal fluid. And they have very little amount of interaction with the neighbors. Whatever they have is what's there in the oviductal fluid, or what is there coming from a few cells, but here they don't have any of that. So there's no cell-cell interaction to worry about, or cell medium interaction to worry about. There's no cytokine involved here. There's no signaling. So if that signaling is not there, then how does this thing differentiate all the way into the blastocyst stage? And it seems like The only thing that is involved here are the two nutrients, pyruvate and glucose. Is this clear? When I say pyruvate, I mean pyruvate lactate, because they keep exchanging with each other. Now, even though glucose is needed for going beyond the 8-cell stage, glycolysis is not. If you didn't get my first slide, then don't worry about this. But the fact is that glucose can be converted into pyruvate by this process of glycolysis, and therefore that pyruvate could be utilized for doing ATP and so on and so forth. But if you block this conversion, it has no effect on formation of blastocyst. In other words, glucose is needed to go beyond the 8-cell stage. It's not needed to go from 1 to 2, 2 to 4, needed for going the first step, but glycolysis is not needed for going down. So what is glucose doing is an interesting question. Okay. Now, there's a few things that I just want to summarize without necessarily showing extensive data for it, although the data will be inherent in the slides that I show. So at this stage, at this very early stage, glycolysis is actually off, which explains why if you inhibit glycolysis, you have no problems growing. So glycolysis is turned off by some inhibitory process. OXPHOS TCA cycle activity, which is what is a reflection of how the Krebs cycle is functioning, is very, very low. In other words, in many different ways, you can show that the membrane potential of the mitochondria is very low in these systems. Why? I don't know why, but it's called the silent embryo hypothesis. And I would assume it's because in many cases, oocytes and early embryos, etc., at a sort of a partial meiotic condition, are held for a large number of years. For humans, it could be held there for 45 years before it's fertilized. And so if there was an extremely high variety of mitochondrion, it will generate a lot of free radicals, and that free radical will cause DNA damage. And so you will have huge amounts of DNA damage and huge amounts of birth defects as a result. So the mitochondria are kept quiet, and glycolysis is off. Now this goes against everything that I just said, which is that you need pyruvate in order to do epigenetic changes and things like that. We know epigenetic changes take place here. And yet, what do you do with that pyruvate? Remember that first slide I showed, there are only two things that can happen with pyruvate. Either glucose converts it and it has two ways to go, or the pyruvate has to come from outside. And in this case, the pyruvate comes from outside. But if that is the case, then how do these metabolites, which I said were essential for changing the proteins which control the epigenetics, how are these things made if you were to not have the pyruvate do anything? So let me handle the glucose thing first, and pyruvate later. The glucose pathway is very interesting. It has glycolysis as the primary pathway, the main spigot, which is open. Then you will make glucose to glucose 6-phosphate all the way up to lactate and pyruvate, as I showed you with glycolysis. But that's not the only way that glucose is utilized. There are other pathways called PPP. It doesn't matter what they are called, but PPP pathway, hexosamine pathway, which come off of glucose metabolism way above where pyruvate is formed. And so when we look at the top part here, these are the intermediates from those earlier steps. When something is red here, that means that if I use a labeled glucose, and this in principle could be done by NMR or whatever, but in reality, we can only do this by isotope log analysis and using a mass spec. And we can take 50 embryos and we can get enough signals. And what you find is that pyruvate and lactate are not labeled by glucose. In other words, glucose is not broken down to make pyruvate or lactate, which goes with what we were saying about blocking glycolysis. So what does glucose do? It's involved in control of these various things, ROS, NAD, pH, etc. But ultimately, it's required for making parts of the nucleotides. So all the ATP, GTP, UTP, AMP, that's what glucose is required for. Now, the other part, which is the hexosamine pathway, it's required for making things like UDP, GluCNAC, etc., which change the glycosylation of proteins. And therefore, they are very useful for many different purposes. You've talked about phosphorylation being an important change in a protein, but glycosylation is just as important. Now, what you find is that these are the ones that are labeled with glucose. And the TCA cycle, which is the Krebs cycle, all of those are labeled with pyruvate. So pyruvate is the way the cell makes energy by running the TCA cycle with the pyruvate that is provided, and not glucose, because not a single molecule of glucose is there in here. So this is a surprising finding, right? I mean, you think that glucose, without glucose, we'll all be dead right away, which we probably will be, but for a different reason. It's not because all the energy is made by glucose. Okay. Now, why without glucose does the embryo block at the 8-cell stage rather than before that? Now, when you think about it, you've got a 2-cell stage, a 4-cell stage where all 4 are identical. And then you get to 8-cell stage. Now there are cells on the outside. This is how the cells divide. So there are cells on the outside. And these cells on the outside have these ICM cells, the stem cells, are on one side of them, and the other side is apical. And whereas the cells which are inside, these cells have got cells surrounding them all around. There is a reason why this edge doesn't do the same thing. But these are both stem cell populations. The stem cell population from here makes the animal, makes the human, makes the mouse, makes whatever, these are the stem cells. But these are also stem cells, and they are known as trophectodermal stem cells. So they make all the stuff that is discarded at birth. So you've got your placenta and yolk sac and so on and so forth, which is discarded at birth, is made from a different population of cells. Now, this is a marker for called CDX2, which marks only the trophectoderm, only the placental precursors. And what you find is that if you were to not have glucose, then these cells no longer express this marker. So in other words, glucose is needed for making these apical cells. Now, what about the stem cell markers? Everyone is interested in those, things, the Yamanaka cocktail markers, OCT4 and NANOG and so on and so forth. When you look at them, they're absolutely not affected. So a stem cell marker not being dependent on glucose and only the precursor for the placenta and so on and so forth is dependent on glucose. How is that even possible? And there are multiple reasons, but one reason is this, which is the PPP pathway that I talked about, which goes off of the glucose pathway. That controls many things, including translation of RNAs. And one of the genes whose translation it controls is called AP2 gamma, which is a transcription factor. Okay? So this is the pathway that is coming from glucose metabolism, but not by the main path. And the HSP pathway, by putting this glycosylation on proteins, then allows the proteins to be present in certain parts of the apical system, and therefore it controls through these molecules, YAP and TEAD, as a transcription factor. Now, what Feng Tao found was that the CDX2 gene, in fact, has a binding site for AP2 gamma and a binding site for YAP-TEAD, and when present together, they synergize the expression of the CDX2 gene. Now, why is this at all important? Because we are used to seeing these kinds of diagrams for signal transduction. We are used to seeing a signaling pathway, like a cytokine, binding a receptor, which then goes and, after a few steps, activates transcription factors, which together on an enhancer will activate a certain gene. Here, that's not the case. Here it's glucose which is activating this particular gene, and it's because of a very intricate system with glucose, where if you turn off the main spigot through which glucose metabolism was going to go, then there is a bigger flux through the side ones, and that flux will cause stabilization of various transcription factors, which together will control a gene. So here is an example of a clear example of a developmental pathway being controlled by glucose and does not necessarily need one of the oncogenic signaling pathways. Okay, so that's quite interesting. Now, getting to the two-cell stage, which I'm more excited about. And so what's the big deal about the two-cell stage? The big deal is that it's probably the most important event in the life of the mouse or the whatever. It's because it's at the two-cell stage mouse embryo, the maternal proteins and RNA, which have been dumped by the mother into the embryo, they all degrade out. The methylation profile is also gone, so that all the gene regulation is gone. And most importantly to our talk today, the genome activation, which is how the embryonic genome takes over, that genome activation process takes place, the major one, at the two-cell stage. So you could not do a genome activation. The embryo will never be able to do a genome activation of its own and has run out of the resources from the mother if this process is blocked at the 2-cell stage. Okay, but where does pyruvate fit into this? Because with pyruvate minus, it's blocked there. Now, I don't know why it took 100 years for people to think about this, but if genome activation happens at the 2-cell stage, and if the absence of pyruvate causes the embryo to block at the 2-cell stage, then isn't it kind of logical to say, is genome activation affected by pyruvate? Which is simple question, again, my kind of question. And so we asked that, and we found that indeed genome activation of the embryo is completely missing. So this is BR-UTP, not the BRDU, but BR-UTP. So this is newly forming RNA measured by this, is completely gone in the absence of pyruvate. The phosphorylation on a particular serine in the polymerase, which is needed for activation of any transcription, that's almost gone. And then there is these epigenetic changes, which people who are experts in epigenetic changes here will recognize are genes which changes which both cause activation and inactivation, and therefore keep a system in a poised kind of a state. So epigenetic changes are affected with pyruvate, but not all of them. Okay? So all the epigenetic changes are not. Okay, now I think I missed, oh, I don't know why I have this again, but okay. What's going on? Okay, thank you so much. Now, how is pyruvate metabolized in this system? Because we are saying that pyruvate is needed for making those epigenetic changes. So how is pyruvate utilized in this system? And this is where things get interesting. So pyruvate is converted by an enzyme complex, specifically by a large subunit or multi-subunit protein called PDH, pyruvate dehydrogenase. Pyruvate dehydrogenase is needed to convert pyruvate into acetyl-CoA eventually. Okay? Now, the way pyruvate dehydrogenase is regulated is through phosphorylation, and except it's the opposite. If it is phosphorylated, then at the kinase, then it is inactive. Okay? And if it is dephosphorylated, then the pyruvate dehydrogenase is active. Pyruvate dehydrogenase is a canonical mitochondrial protein which is folded in the mitochondrion. It's supposed to be in the mitochondrion, always is in the mitochondrion. And we used an antibody which recognizes phospho-PDH, and that's the inactive version. And you can see that what I was saying was correct, which is the mitochondria are very low in their activity because all of these are staining in green, which is against the phospho group of phosphopyruvate, of phospho-PDH. But the interesting thing is, if you use an antibody which is not dependent on phosphorylation, which is pan-PDH, now you find that in red here, and these are actually yellow, as you can see. So there's a little bit of that here, but most of the active PDH is in the nucleus. Okay, now if you wrote this in your MSc exams, you'll immediately get a 0 for it, because who has ever heard of a molecule like PDH, which is a bona fide mitochondrial protein, and realize mitochondria has 2 membranes, and then the nucleus has 2 membranes, and you are transferring a mitochondrial protein into the nucleus. Now, let's say, for argument's sake, that it somehow gets into the nucleus. In fact, in some cancers, people had shown that it might go into the nucleus. But even if PDH goes into the nucleus and makes acetyl-CoA, that's going to be the end of it. There is no way the TCA cycle, which you have all learned since 7th grade and all forgotten, guaranteed, is run by these enzymes, citrate synthase, isocitrate synthase, alpha-ketoglutarate. All of these things are mitochondrial proteins. So this is bona fide mitochondrial work where the TCA cycle works. And so even if you had acetyl-CoA, okay, so you might make citrate and that'll be it. But remember that for epigenetic changes, you not only need citrate, but you need isocitrate, which can give rise to NAD+, which is very important for this process. Alpha-ketoglutarate, I mentioned, is needed for methylation and so on and so forth. So you do need these as well. And so what's going on? We looked at aconitase, the very first enzyme, bona fide mitochondrial protein, and you find that aconitase, the TCA cycle protein, is in the nucleus. And it's in the nucleus in a pyruvate-dependent manner. So if you leave out the pyruvate, it cannot get into the nucleus. Now, if you do this for other proteins, why? Okay, if you look at it for other proteins in the TCA cycle, you find that these guys, this is the one that I showed you, and then there's PCB, aconitase, and IDH3A, etc. These proteins are all in the nucleus at this point of time. They are never in the nucleus at any other stage of their life. They are always mitochondrial, as they should be, but they are transferred into the nucleus. This one, KDGH, it's a postdoc-dependent expression pattern. Sometimes you find it, sometimes you don't. But it's absolutely clear that these last ones, the malate dehydrogenase and so on and so forth, these are not found in the nucleus. So succinate dehydrogenase, malate dehydrogenase are not found in the nucleus. So it's a not only strange thing that they are found in the nucleus in the first place, but even stranger that it's not all of them, but a specific subset. Okay, now this is the TCA cycle again, and what you find is that this is not random. This protein, this protein, this protein, these are all, this a little bit, are inserted into the nucleus at this point, and these guys are not inserted into the nucleus, which makes a lot of sense, and I'm not going into the data, but we have done assays that tell us that these enzymes are actually active inside the nucleus. So these active enzymes inside the nucleus will now make citrate, isocitrate, alpha-ketoglutarate, everything that is needed for epigenetic changes. And it's a good idea not to have these guys in there, because things like succinate and malate, they not only inhibit some of these earlier enzymes, but they also, by themselves, cause increase in ROS levels. So you don't want the free radicals to go up. So, but the system is just totally amazing, where this half is moved into the nucleus and that half is not. So the so-called Krebs cycle, and don't call it Krebs cycle if you are in Hungary, in Szeged in particular, because the people there worship Saint Georgi, who found this half of the Krebs cycle. I mean, calling anything negative about Saint Georgi is like saying something bad about Tagore in Kolkata. And so this is, this part is the ones that go into the nucleus, and that part was found by Krebs. Krebs completed the cycle, and then it became the citric acid cycle, okay, or TCA. All right, so now when we find these guys in the nucleus but not those, and there's a lot other reasons that I could quote, but I don't have time for it, which makes me think, well, first of all, that why was Saint Georgi so interested in citrate? Probably because the Hungarian red peppers with which goulash is made has got a lot of citrate. I made that up. I don't think that's the real reason. But more importantly, why did two different guys find the two sides? I think because the Krebs cycle or the TCA cycle or whatever you want to call it is not a cycle, period. You say that again in your exams and you're done for. But the Krebs cycle is not a cycle. Whenever the Krebs cycle is a combination of two half cycles. Okay, this is one half cycle, and that's the other half cycle. If you feed a cell, starve a cell, do whatever, refeeding a cell, all of these guys behave in the same manner, and all of those guys behave in a different way. So what happens is, in a normal cell where nothing is lacking, the Krebs cycle is a cycle. It goes round and round and round. But as soon as you have a stress situation or cancer, or I mean, we've seen it in cancer cells, and other people have too by now, and or some severe stress, then it doesn't ever break here or break there. It breaks over here. So all of these are controlled in the same way. All of those are controlled in the same way. And so under stress conditions, the Krebs cycle, the TCA cycle is not a cycle. They are not in equilibrium with each other. It's only under completely normal conditions that they are in equilibrium, and then the cycle can go round and round. Okay, and so in this case, the presence of these enzymes in the nucleus means that those enzymes are functional inside the nucleus and can make the proper metabolites that you need, and those guys are completely dissociated from it because they never make it into the nucleus. Okay, so now, we used to work on flies, and then people said, why don't you work on mouse? And then we worked on mouse, and then people said, why aren't you working on humans? I mean, this is endless, right? So what happens in humans? What happens in human embryos? The problem is you really cannot ethically use a lot of human embryos. There are IVF clinics with a lot of proper paperwork. You can get discarded embryos, human embryos, and we had 5, and that's it. This is n equals 1 for each, 5 embryos. Okay, and this we stained with the same antibody, the one that recognizes nuclear versus the non-nuclear, or the inactive versus the active. And this is a single cell human embryo. And you see what you expect to see. The cell has got mitochondria which are inactive. That's what I said about a silent embryo hypothesis. It's fertilized egg, but it has inactive mitochondria. Okay? Now, when they start, these are the two pronuclei, one from the male, one from the female. When they start fusing, now you still see the same thing. Very distressingly though, at the 2-cell stage, where in mouse, we had seen nuclear localization of this protein, and that to the active form of this protein, we saw nothing in the nucleus. And by we, I mean Raghavendra, who was about to have a heart attack, because now this was going to, paper was going to get stuck, it'll never get into cell, because it works for mouse, but it doesn't work for embryo. Okay, all of that, and it takes more than a week to do each step. And then this is our last embryo, which is, well, these two are the last two, 4-cell stage and 8-cell stage. And you find that as soon as you get to the 4-cell stage, you see nuclear localization of PDH and active PDH. At the 8-cell stage, you actually have more, the picture doesn't do justice, but you have the highest amount quantitatively. And then this cell was a little bit ahead of the others. And what you find is as soon as it goes ahead, it loses completely. And this is the phenomenon. At the earliest stages of development, the mitochondrial proteins go into the nucleus so that you can have the epigenetic modifications that you need for embryonic genome activation. And once embryo genome has been reorganized, the biggest reorganization that you will need in life, once that has happened, now they are never seen in the nucleus unless there is a major stress like cancer or like some other stress which I don't know about. I'm sure will probably do the same thing. Okay. So, why? Anyone, any student wants to say why? Yeah? Did someone raise their hands? Why is it that it does go into the nucleus, but it goes into the nucleus at the wrong time? Yeah? The answer is right here, which is that the human and mouse embryos go through genome activation at different times. So the mouse embryo goes through genome activation at the 2-cell stage, and the human embryo goes through genome activation at the 4 to 8-cell stage. So in spite of its being a result with just n equals 1, I couldn't have made this up. We know that the genome activation in humans is at this point, and it is at exactly that point that the PDH is going into the nucleus and is doing the same thing as in mouse, but it doesn't need to do that at the 2-cell stage. It needs to do that at the 4 to 8-cell stage. So this was actually a very satisfying result that it was delayed. Now, so in my mind, I showed this slide before showing that all these metabolites are made in the mitochondrion. And in my mind, this is the ultimate example of, mountain coming to Muhammad. There is, the mitochondria are all inactive. So there is no way to run the TCA cycle in the mitochondrion in order to generate, various things, including energy, ATP, etc. So what does the system do? It moves the mitochondrial proteins that are needed for genome activation into the nucleus. and there they perform the function they were supposed to perform in the, and if I may just for the students say this, that if you start to work on a problem where you know absolutely nothing about the problem, but you want to solve a problem. You don't want to just say, so-and-so in 1981 or 2001 showed this, and therefore I'm going to see whether in my cells this happens or not. You want to say, this is a phenomenon I want to understand. Then new things start coming up. I mean, this was not something that one would have predicted, and it's nice when it's a new inquiry-driven finding. It's only for the students. The faculty know that. Okay, now that's well and good. How does a protein like this go into the nucleus? This is a bar that shows you that the PDH complex is bigger than a ribosome. Now, this PDH complex, bigger than a ribosome, is sitting inside the membrane of the mitochondrion, somehow making it through two membranes, crossing two membranes of the nucleus, and ending up inside the nucleus. How can you possibly do this? Now, one of the ways in which large proteins can get in without necessarily having a nuclear localization signal is through nuclear pores. And here is another unexpected result that you would not, nobody knew about, but which is, if I stay now with nucleoporins, or it's a complex, they should be a band around, a ring around the nucleus. But at this stage, they are not. They are present in little dots all over the cytoplasm. Now, whoever has heard of nucleoporins, they are made in the cytoplasm, of course, but they immediately get into the nuclear periphery because they have to transport things into the nucleus. But they are all instead along in little dots like that. And at this point of time, the inner membrane is formed, but the outer membrane of the nucleus is just about being formed. Okay, the outer membrane is an extension of the ER and so on and so forth. So these dots actually are coincident with ER markers and some Golgi markers as well. Now, okay, so they're there, and it's possible that somehow nucleoporin is involved in that. But still, there is a problem here, which is how is it that they would ever get into the nucleus? And when you look at the staining pattern of NUPs and mitochondria, so this is nucleoporins and mitochondria, you find that rather than being on the nuclear periphery, in fact, these mitochondria, which is in red, have NUPs on them, which is absolutely mind-boggling, but they do have the nucleoporin protein attached to them. Okay, these make me sick. Now, I'm not going to go through this. The time is getting late. So I'm just going to tell you this is how they move. They move to the nucleus the same way that a mitochondrion moves in the neuron. In the neuron, the mitochondrion moves from the synaptic areas to the cell body, and everything about this is taken from there. So the mitochondrion is attached by these proteins, Milton, into kinesin, and then the bundles of acetylated tubulin on which they move and they get to the nucleus. Doesn't answer the question how the protein gets into the nucleus, but at least the mitochondrion can move like this towards the nucleus. I'm going to skip this slide. Now, the answer comes from seeing how a protein which is inserted into the nucleus, like PDH that we talked about, versus SDH, the succinate dehydrogenase, which was on the other side of the TCA cycle, which does not enter the mitochondrion. And you do a double staining to see what happens. And when you do that double staining, you see this really interesting thing. Indeed, SDHA does not enter the nucleus. You see a lot of PDH in the nucleus over here. But then you see these little dots. I hope they're visible in the back, but there are little dots which start off on the mitochondrion, and then they become independent of the mitochondria. So what are these little dots? They are positive for PDH, and if we forget about the pyruvate, then we don't see the little dots anymore. Okay, so somehow these guys are getting into these little dots, and indeed, one of the experts on this in India is Ananta Lakshmi, who's at Trivandrum in the other institute, Rajiv Gandhi. And so I called her up from the US and said, you know, what do you think? And she gave me the dimensions and how it should be for mitochondrion-derived vesicles. And so these are mitochondria-derived vesicles, and they are budding off. They are budding off from the mitochondrion. How do I know that? Because, again, from her work as well as Heidi McBride's work, we know that at those junctions, the DRP protein, which is the fission protein, which separates the mitochondria into two halves, at this, when they form these vesicles, then DRP forms a little ring around the vesicles. DRP is retained, but the vesicles separate out after being cut. So, fortunately from Heidi McBride's laboratory, we knew approximately what is going on. And that's because when a mitochondrion is really old, it needs its components recycled. It then creates these vesicles, and then MDVs they are called. It has cargo proteins, sends them to late endosomes, lysosomes, whatever, and in order to recycle the proteins. Now, we don't have very old mitochondria. What we have are very new mitochondria. And like very old mitochondria, very new mitochondria are not active either. And so as a result, they are prone to forming these vesicles which move along tubulin. And why do they go into the nucleus is because the NUP complex, the nucleoporin complex, is attached to them. So where they go, whether they go to the peroxisome, lysosome, or the nucleus, is determined by a cargo that is targeted towards that. So the NUP complex goes and forms the same mitochondrial barrier just around the nucleus, from which then they are able to get into the nucleus without necessarily crossing two membranes. So the outer membrane is just being formed by the ER. The ER is attached to this, and it either fuses or uses the NUP, we don't know which one, to throw these large proteins into the nucleus. Okay, I had two quick things to say without any data, but you know, if it's getting late, please feel free to leave. But they are important kind of basic observations. One is that when we talk about metabolism, we have to understand that metabolism is not a static thing. It is very flexible. Depending upon what you give a cell, it behaves in such a manner. So at the beginning, you needed pyruvate. You just could not do without pyruvate. Without pyruvate, it get a 2-cell block. Later on, you need glucose at the 8-cell stage. So you have now made the cell a little more flexible. And even later, at 82 hours, you don't need anything. You get rid of everything except salt and buffer, and you find that it still goes into the blastocyst stage. How would a cell an embryo do that? How will it survive? The answer to that, and well, the reason to that is a rationalization, is that by this time, the embryo has left the oviductal fluid, has now moved into the uterus, and is trying to get implanted with the real energy source, which is the mother's blood. At that point of time, there is no nutrient around, very little, I mean, it's migration. And yet it does that. How does it do that? It makes whatever amount of fatty acids it made in the first rounds, it now utilizes that fatty acid in order to burn it and derive energy from it to survive here. So any strategy that you can think of, nature has thought of that in advance. The second point, quick point I want to make is people all the time want to look for differences. Oh, no, this is different, that is different. I work on stem cells, and stem cells are different from cancer cells. Cancer cells are different from embryonic cells, developmental cells. Coming from Drosophila, I just can't believe that the same thing will keep chasing us here. And it's the similarities that matter. The embryo requires pyruvate in order to form. The stem cell requires glucose to form, and cancer cells require glutamine to form. But nothing is sacrosanct here. That's what's available and is programmed for, and is utilized depending upon the redox balance of the cell. I can change the redox balance, the NADH to NAD plus ratios, in such a way that this cell will utilize glucose rather than pyruvate. But the cancer cells, for sure, in a petri dish where you have added a bunch of glutamine, they do become glutamine dependent, but in vivo they are cancers which are pyruvate dependent, and so on and so forth. So the idea is just simple, that metabolism doesn't get reinvented every time you come up with a new cell type or something like that. What happens is how you utilize the metabolism with minor tweaking of parameters that determines nutrient utilization. I've already said this work was done by Raghavendra, Mark, and Feng Tao, and so there's 3 papers on this, and then that's it. They've all contributed to each one of them, and this was made possible by the NIH Director's Pioneer Award. Thank you very much.
Thank you, Professor Banerjee, for a wonderful talk. So now time for questions. Quick questions.
Yeah.
The question here.
Sir, it's a wonderful
Raise
talk.
your hand with the microphone and wave it a little bit so my visual cortex can sense the motion. Yeah, okay.
Thank you, sir. It's a wonderful talk. I just want to know about those proteins which are going to the inside the nucleus at the 2-cell stage, or at the 4 cell stage, whether they will get degraded inside the nucleus or
Yeah.
they will come out from there.
Yeah, they probably
What is the
will.
fate actually?
Yeah, they probably will. I mean, it's almost impossible for them to then go back into the mitochondria. There is no way.
So mitochondria
when-
will, nucleus will again regenerate those proteins
Yeah,
from mitochondria.
so it will either get degraded in the nucleus where there are lots of ubiquitination pathways, or it'll get excreted into the cytoplasm and get degraded. Whichever it is, we don't see them anymore. You know, if you just stay in a natural cell, any cell other than these very specific ones, you never see them.
Thank you.
Yeah.
So
Yeah.
you mentioned that the first half of the Krebs cycle is taking place inside the nucleus. So how do redox balancing happen? Because NADH has to be oxidized back to NAD. So that happens in the mitochondrial electron transport chain.
Yes and no. I should probably just, if there's time, I should probably just answer this question a little bit later for you.
Okay.
Because it's not a simple answer. But very quickly, lactate dehydrogenase levels are extremely high before the two-cell stage. And therefore, it causes a huge amount of a change in this lactate to pyruvate conversion. Okay, so any pyruvate that might be inside will get transported out and become lactate. Okay, now the Krebs cycle, it is able to control these as well, and so does the ETC, as you pointed out. But these are controlled in very different ways. NAD plus is actually manufactured there, and what happens is that the cell is under enormous amounts of reductive stress. So when you have that much of NADH there, then all the lactate gets converted to pyruvate. All the pyruvate gets converted to lactate, and the reverse reaction does not happen. Okay. Now we can override that by fooling the system and adding something which is not pyruvate, but a derivative of pyruvate. Now that thing cannot use lactate dehydrogenase A. It uses another enzyme, but it rebalances the NADH, NAD+. So it's a matter of the cytoplasmic NAD, NADH balance, which is somehow doing this. And the LDH levels, which were in kilogram quantities before this, right after the genome activation, it drops to pretty much zero. So now there's none of this LDH left anymore.
Thank you.
Very good talk.
Yeah.
In the initial part of your talk, you mentioned about the minimal media, wherein you can avoid amino acids. How do these mammalian cells can make essential amino acids from the minimal media?
Oh boy. Okay, it's not going to cooperate. This is a really important and interesting point. Could you go to my second slide, please? No, forward. More. Yes. No, that one. When we add either glucose or pyruvate, remember that's the red and the blue, which tells us what gets labeled by glucose and what gets labeled by pyruvate, right? In order to save time, I did not talk about this, which is there's a bunch of things which don't get labeled by anything. So external pyruvate, external glucose does not label any amino acid except for these which are made by the TCA cycle. Okay? Now, you're absolutely valid asking where does the cell get these from? And that's why the mother puts in the amount of protein into an embryo as she does. So these proteins essentially act as what would be called yolk, but they're protein based, and these proteins are all degraded inside the cell to generate all of these amino acids, all of these nucleobases, fatty acids, and so on and so forth. This thing is a machine. It has everything that it needs except for this little sort of glucose and pyruvate machinery to provide from outside. Okay? It would be so nice for some engineer to say, okay, I'm going to make something which actually follows this whole process. Yeah.
All right, so maybe in the interest of time, we can take all further questions offline. So for this wonderful talk, as a token of our deep appreciation, I would request our Director, Professor Jain Murthy, to hand over a small memento to Professor Utpal Banerjee.
Oh my goodness.
Right?
I have one.
You have one?
From
Can I keep it for myself?
You're welcome. I got exactly,
Then we'll witness it, yeah?
I got exactly this from the Rajiv Gandhi Center yesterday. and this Padmanabha is
This you won't have got.
really
It is different.
heavy. Okay. Yeah, I'll keep that too.
Yeah, okay.
Yeah. Please don't.
I was going to keep
Yeah,
myself.
no. You're the director. You can get one made. All right. Thank you so much. Thank you, sir.
Thank
you.
you, Professor Murthy. Thank you, Professor Banerjee, for this wonderful talk. Thanks, everyone, for your rapt attention. And I'm sure students would have additional questions. So at least for a little while, if you want to come up here or ask him, talk to him, it's fine.
It's okay. Huh?
It will. I will get it.
What do you think?